Hi, everyone. I'm Natasha from Carnegie, and I'm here with Dr. Nipam Patel, who is the director of the Marine Biological Laboratory and also a professor at the University of Chicago. He is speaking tonight at the Carnegie Department of Embryology about his very exciting work. So maybe to start out, if you could just give us a little quick elevator pitch of what it is that you do.
Right. So I'm essentially a developmental biologist who's very interested in the evolution of development, both in trying to understand how diversity is generated, so how organisms become so different from one another despite the fact that there's a lot of conservation in their genome, and how developmental mechanisms evolved. And I've done that in a whole bunch of different organisms over my career, and I'm interested in continuing that, understanding both the evolution of diversity of morphology, and more recently also interested in understanding generating diversity in color in butterflies.
That is so cool. I mean, I'm so excited for this talk. I wish it was happening right now. I'm sure you're glad to have a couple hours.
Yeah.
But and so a very general question, just over the course of your career, looking back to when you maybe were a grad student through today, what would you say are the most exciting developments that have occurred in your field?
Yeah. Well, I think one of the great excitements, I think early on back when I was just starting as an undergraduate was really the time that there was this explosion of using a genetic approach to understanding development, to dissecting development, using genetics. And a lot of that was the leadership in that was in the fields of C. elegans and Drosophila as those sort of emerging organisms that you could take a forward genetic approach. And I'd had classical training in embryology. I even had an embryology course as a high school student, but it was I was really fortunate, but it was very descriptive. But there were, some really exciting experiments that had been done that helped us understand the mechanisms. But I think at least, for a lot of the thinking now, as a big breakthrough came when people started applying genetic approaches to really understanding what the genes were to get down to that level of understanding. And that really opened up the field of developmental biology in a really new way. And then I was really fortunate that as a grad student, then was the time that people started saying, well, now that we understand this kind of detail about development, maybe we can understand how development evolves. And I had sort of made an somewhat accidental discovery when I was somewhat accidental. Yeah. Well, this one was definitely just doing some experiments and then realizing something and realizing that I could now answer a question that a lot of people were really interested in, but had technically been having a hard time making headway, and now I could actually sort of rapidly start to gather data. And that, I was very excited. I was fortunate to have a mentor that was really happy to let me go and do that, to switch my entire PhD project and do something completely different, actually. And that was great for me, even though what I'd been doing up to that point was quite successful. I switched over to something else that, fortunately, I was also very successful at. And I became very excited about this approach of trying to understand the evolution of development. Because before that, I'd really been mostly focused on the development of specific species and not really thought about it from an evolutionary viewpoint. But then there's all this wonderful literature that was out there about why these were interesting questions and what some of the questions really were. And I think then, then developing new organisms that we could study for that reason. And that was a long road to go through, because trying to bring them up to the level of the organisms, the research organisms that we primarily use, took quite a lot of time. But I think over especially the last 5 years, there's been great excitement now, because a lot of those tools that have been developed in the standard systems now become rapidly applied to the new organisms that we're working on. Starting with just techniques for looking at gene expression, and then moving on to doing things like RNAi, and then now using gene editing and things like that. It's really opened up the field enormously, combined with being able to sequence genomes and all of that kind of stuff.
So it makes each new model sort of a little easier to bring on board.
Absolutely, to bring on board and to really generate results that you can are really meaningful. Yeah.
Right. And so then, that's kind of looking backwards or over the course of your career. What about looking forwards? What advancements are you anticipating are going to happen over the next 10, 20 years that you think will be, breakthrough.
Yeah, well, I think some of the things now are, again, advancements in genome engineering, the ease with which you can do all sorts of experiments. And in fact, it's even almost dangerous to kind of predict, because in so many of the systems now, there's just constantly breakthroughs in the technology. And I think we're finding them easier and easier to apply to a greater and greater number of organisms. And then, so genome editing, and then also imaging technologies. So I think one of the great things in developmental biology is really being able to see events unfold live before your eyes and not just looking them as static things. I think there's great utility in that. And I think just in general, as we learn to work with more and more organisms, one of the ways I like to couch it is that, there's a lot of really hard and interesting problems in biology, but that evolution has actually already solved many of them. And it's just a matter of finding the organisms that have solved those problems and then making them tractable to understand what was the solution that the organism came up with. Why does it, succeed in this? Good example, are organisms that can survive extreme environments, right? So there are plenty of organisms that already figured out how to do that, but they're often not ones that we work on. But now they're more and more experimentally tractable. The same thing drove me with the structural coloration in butterflies. Physicists understand it very well to the point where they can write out the equations and tell you exactly how it works. But we have no idea how a living cell makes some of those things. But now we have the technology, both from imaging and being able to manipulate the organisms, to now really get fundamental insights into how those things work. The other organism that we work on in the lab is a crustacean called Parhyala. And actually my start in being interested in crustaceans really started at the Carnegie when I was a staff associate. Up until that point, I had done all my comparative work in insects. And again, it partly comes from, I had a hobby of diving, and I got to know the curator of the crustacean collection when I was here, because he's obviously just in Washington, D.C., and got to talking about some of the really exciting questions that we get asked by branching out into other arthropods, into crustaceans. And I was really fortunate that I got to go to this little island off the coast of Belize to go collect animals when I was here at the Carnegie, and that really got me started to working on them. And then after I left here, I was initially at the University of Chicago, and we started developing an experimental system where now it's really a very tractable system where we can do gene knockouts and make transgenic animals, but really get at fundamental answers to how body plans are set up. So we study how you get the right appendage on the right segment. And now we have a pretty solid understanding of a lot of that. And from that, we can understand how you evolve that system to make other crustacean body plans. But I think the excitement now is that we can increasingly get more and more of those organisms and get somewhere with them. So for Parhyala, the crustacean we work on, it took us something like 20 years to get to where we are now of working on it. But I think that if, given the tools that we have now, to redo everything that we did would take probably 3 or 4 years. And
That's
so,
incredible.
so I think it's a really wonderful opportunity now to really expand the repertoire of organisms that are worked on.
That's blowing my mind. Just thinking about that time frame shrinking that much in not that long not that many years.
Yeah, well, for example, the animal, the Parhyala, it has a genome that's about 10% larger than a human genome. So, when it became possible to sequence genomes, well, no one was ever going to do Parhyala because it was going to be cost, as much as a human genome or more. But now the costs are so low that you could say, okay, we'll just do it.
Go for it.
Yeah.
Right. That's really interesting. So I wanted your talk tonight is called The Physics of Beauty, and it's about butterfly coloration. And one of the things that struck me, especially as we were preparing to, market this to the audiences, is that because there's such great visual appeal to this subject, that maybe there's an opportunity to reach people in the general public who maybe don't think of themselves as interested in science, but could be hooked in by sort of the visual beauty of butterflies. And do you see that as an opportunity that butterfly research has?
Yeah, absolutely. I think that's actually in general something that's really excellent about developmental biology, is that it is very visual. I think you can show movies of developing embryos and people will instantly sort of connect and understand that there's something really exciting happening there, and trying to understand how it happens. So for example, I've been doing a couple of different public outreach kinds of things. And some of them end up quite by accident. Like I posted a movie of frog development. It was just a movie I happened to take of going from a one-cell embryo into a hatching tadpole. I mean, it worked, it's a pretty nice movie. Everything stays in focus and everything. And I posted it, and, a few people saw it, and then it got posted by someone on a public site in Japan who said, well, it reminded him of the tadpoles in the pond, in the rice paddies when he was growing up. And the next thing you know, it had millions of views.
Wow.
And people were all excited. And in that movie, 2 embryos die. And there were literally thousands of people who wrote in things asking, well, why did these embryos die? What's happening? And that's a great opportunity, right, to really talk about and connect with people. And I think, like I said, that's one of the great things about developmental biology. But yeah, the butterflies are another one. That's a very charismatic group of organisms. Everyone can kind of relate to them in some way.
Doesn't like butterflies.
Yeah, and it is, and as you'll see, hopefully, from the talk, is that it is a fascinating sort of question about how they generate color. That you can, again, because it's very visual, people will understand right away the problem that's there. And then you can explain, I think, in relatively simple terms, even though you are talking about physics, that it's basic enough physics that you can walk people through how the system works and how complicated it is to think about how a cell would actually make what it needs to do to make that function.
Right, but having that beautiful wing to look at and then walk backwards into the physics and the biology is such a great way to draw people in.
Yeah, absolutely. And I think scientists have to get better and better about connecting to the public, to really, for the public to understand the importance of the work we do, even if it means, starting with something that's kind of eye candy and then working our way through, especially because it's one thing to talk about working on diseases and people get that, but so many of the huge breakthroughs in biology come from people doing very, very basic research. Absolutely, and people often forget that, and certainly funding agencies seem to be able to forget that very often. But I think engage. But partly that's on us as scientists to really make the public understand that this kind of research we do, we can't even always know what kind of breakthroughs it's going to lead to. In fact, I would say that often we don't know what kind of breakthroughs it's going to lead to.
It's the things that you don't even know to get excited about that end up being the most exciting.
Often, yeah.
And you kind of teed up my very next question perfectly, which is just, there seems to be an increasing awareness that scientists should get better at communicating with the public, and clearly it's something that you are good at. And what tips would you give to peers who recognize this is something that they need to get better at, but kind of don't know how to start?
Yeah, so I think like when I even work with my own students and postdocs and things about thinking about giving talks, it's, I think one of the things, especially in engaging the public, is to really talk as though you're having a conversation and telling a story. I've often seen people who are giving a talk as though they're writing a paper, and that does not appeal to people. There's a reason that you have science papers organized the way they are, but that's not how you would talk to someone.
Right.
And so I think that that's very important to think about, both from the communication side and also even the visual side, that engaging people is very different than figures in a paper and things like that, and to think that through. And I think that I've had colleagues tell me, after a talk, and they say, well, one of the things that appeals is that what you really do is you tell stories. And I think that that's an important thing to do, especially when you're talking to the public. I would say that's even an important thing to do when you're talking to other scientists.
I agree.
Yeah.
And that I think, especially talking to the public, not only does the story hook them in and engage them with the science, but it also makes them think about scientists as human beings, which I think so often people just think of the scientist as this removed figure who spouts facts and figures, but isn't really very human or personal. And so,
well, as I'll mention today, right, the whole work with the butterfly started when I was 8 years old, and I found a dead butterfly in my yard, and then read a book on how to mount it, and then convinced my mom to sew me a butterfly net, and and now I have a huge collection, and from that I was always very fascinated in biology. I had a great conversation with Joe Gall about how, he was an insect and butterfly collector as well, and that's one of the things that got him really excited about science and biology. And I think, it's true for a lot of young kids that it's those kinds of things in the outdoor world. And now it's great as a scientist that you can come back to that love, but actually apply the tools that you have now to making discoveries.
It's like a synthesis of your childhood passion and your scientific
training,
knowledge and training. And then I wanted to ask you a question from your role as a director, do you think that, it seems to me that interdisciplinary science is increasingly important, and do you agree with that, and why do you think that's the case?
Yeah, no, I mean, I think as we get more and more into trying to get answers to questions, we realize we need to bring in tools and knowledge from lots of different areas. And the question is, do you do that by trying to learn all those different areas, or do you do it by collaboration? Obviously, a bit of both is what you want to do. But now things are, certain areas are so complicated that it's really hard to be a master in all of these things. And so I think what's really important now is to actually have at least a knowledge of what those different areas can do for you, and then to seek out people who you can interact well with and communicate well with, and understand what the important questions are and work together to answer those. So I think you see a lot of good examples, especially now with like computational approaches that, that I think everyone should have an appreciation for what computation can do, but all of us are going to have different levels at which we can do it. And there are people who are really good at that, who then, would be great to, or are really great to interact with, but they need the biologist to really say, well, this is the question that I want to answer. And and I think that you see the same thing with the interaction between biologists and chemists, or different areas of biology and physics and so on.
Yeah, you kind of need those different viewpoints to even think of the questions that you want to ask, and then how to pursue the answers.
Yeah, both the questions to ask, and then also knowing which are answerable right now.
Which are a dead end.
Or well, or just right now that doesn't exist in an approach.
Capable or whatever. And then I had one last question. You mentioned earlier about how you had been a staff associate here at Carnegie Embryology, and so I was wondering if you could just talk about the role Carnegie has played in your
a huge role. So I was here from 1990 to 1994. I was a staff associate, and it was one of those situations where I came right out of my PhD, and I had a good idea of what I wanted to do and something new and interesting that I was working on, and this place just really gave me the freedom to run with it. And to go in all different directions. And it was just an incredible environment for doing it, where people were extremely supportive and very interactive, and I could go to anyone and talk and get advice. But in the end, it was my own work. It was up to me to do what I wanted to do. And I was really fortunate. It was just, it was me and a research technician, and just plowing away in the lab, not having other distractions, and really having the freedom to do what I wanted to do. And it was just really, really great fun, and exploring and getting to do things. And it's had a huge impact on my career, both in exactly what I do. Like I said, I started working on the crustacean systems when I was here. And then also really just indirectly, in the sense that the whole idea of what it means to be a mentor, how people should do science, and the real value in being creative and being kind of fearless to do what you want to do.
I love that. I mean, creativity in science is something that I feel like we talk about every day here, practically, and so I like hearing that was a formative part of your experience.
Absolutely, yeah. It was wonderful 4 years, so. And I always like coming back here. It's just a wonderful place to visit and seeing all the things that are going on.
Well, we're so happy to have you back. Thank you so much, and we're very excited for your talk tonight. I know it's going to be excellent.
All right.
So thank you so much.
Thanks.